Hi, let's talk about some tricky concepts related to the immune system. I'm in chapter 11 now. First of all, let's talk about innate immunity, the so-called first line of defense. The truth is, innate immunity and the specific or acquired immune responses don't work entirely independently. But generally speaking, innate immune responses are the first responses to a bacterial or viral infection. They're called innate because they don't rely on specific recognition of the virus or the bacteria. That is, these responses will occur regardless of which particular bacterium or which particular virus gets into the body. Perhaps a hallmark of the innate immune response is the inflammatory response. There's a great diagram. It's figure 1114 that summarizes the inflammatory response. Generally occurs when there is a bacterial infection, introduction of a bacterium into the body. But this type of inflammatory response is also seen with injury. Say you fall on your knee, bang your elbow on something and it swells and it hurts. That too is an inflammatory response to the tissue damage. So inflammation is a general response. Here, inflammation is the innate response to a bacterial infection. And inflammation is initiated, with regards to bacterial infection, is initiated by the introduction of the bacterium below the surface of the skin. And the primary players in the inflammatory response are the phagocytes. First, the resident macrophages. They're called resident because they're always living there below the surface of the skin, just kind of waiting for a bacterial invasion. So it's those phagocytes, the resident macrophages, and a very powerful type of phagocyte that has to be recruited to the area, the neutrophils. So these are the main players with regard to containing and destroying the bacterium at the site. And that's really the goal of the inflammatory response, is to prevent the bacterial infection from getting much further than that initial site of introduction. So, bacteria is introduced below the surface of the skin. You step on a rusty nail, sliver under the skin, something like that. And it's those resident macrophages start gobbling up the bacteria. Again, it doesn't matter which bacterium it is, they're just going to start gobbling them up. And when they do this, when they start engulfing the bacteria, the macrophages release chemicals. And these chemicals, these cytokines, recruit other cells to the area. These chemicals work in a couple different ways. First of all, they can activate mast cells. And these mast cells themselves start releasing chemicals. And those chemicals, notably histamine, dilate arterioles, make capillaries really leaky, and they're setting the stage to make it easier for phagocytic specialists like the neutrophils to arrive onto the scene. They arrive onto the scene, they enter these leaky capillaries, these neutrophils circulating in the bloodstream, enter the leaky capillaries and they squeeze out through those pores that have been made just a little bit bigger. And these neutrophils are now very powerful phagocytes. They show up and gobble up and contain the bacterium and hopefully keep it from spreading too much further than that initial site of infection. Now, you know, bacterial infections, even minor ones, can produce a little kind of nasty pus below the skin. All that white stuff that's in the pus is the white blood cells and the bacteria that they've eaten. That's literally why it's white, because of the white cells, notably neutrophils, that have arrived at the scene to eat up those bacteria. There are a couple other aspects to innate immunity beyond just the inflammatory response. The inflammation is one aspect of innate immunity. The other aspects of innate immunity include interferon, complement protein, and natural killer cells. Interferon is a chemical that's released by virally infected cells. And again, it doesn't matter which virus gets into the cells. The cells respond to this viral infection by releasing chemical interferon. And interferon blocks the replication machinery of neighboring cells. So it takes out a couple of the neighbors. They're not going to be able to replicate. But that means the virus isn't going to be able to use that replication machinery either. So interferon, a part of the innate immune response. Complement proteins, these are interesting proteins. They circulate in the plasma as independent proteins, and when they sneak out of the leaky capillaries, they form into these membrane attacks complex. That is, these proteins arrange themselves in a very specific fashion, insert themselves into the wall of the bacteria, and they form big pores in the bacterial wall. These pores allow water to rapidly enter the cell, and the cells burst under the pressure of all that water. Lastly, with regard to innate immune response, are the natural killer cells. Natural killer cells are kind of an interesting cell type, almost like a lymphocyte, but they're not. We don't know exactly the origin. They're not quite lymphocytes, but natural killer cells are really good at gobbling up virally infected cells. So those are the four components of the innate immune response. Again, so called because this response occurs regardless of the specific bacterial or viral infection. On the other hand, the acquired or adaptive immune responses are all about the lymphocytes, B cells and T cells, responding to specific bacteria or specific viruses that get into the body. And the way they do this is by the presence of antigen receptors. Both B and T cells have specific receptors that we call antigen receptors. Now remember, an antigen is a very large molecule that's on the surface of a bacterium or a virus. And this is specifically what the body responds to. Those molecules are specifically what the B and T cells respond to. Not the entire virus or the entire bacterium, but a molecule that's displayed on the surface of the bacterium or the virus. And when that molecule, that antigen, comes in contact with a receptor that it can bind to really well, it activates the B cell or the T cell. So the receptors on the B cells and the T cells are very, very specific. For the thousands and thousands of types of antigens out there, there are thousands and thousands of types of receptors. Thus, there's thousands and thousands of types of B cells and T cells. So it's a very specific response. When B cells encounter an antigen that fits their receptor well, that particular population of B cells, and not the other ones, but only the ones whose receptor binds well with the antigen, that population of B cells undergoes a very rapid expansion that we call clonal expansion. Those B cells replicate very quickly, and they become protein factories. Most of those cells that are produced in clonal expansion start cranking out antibodies. So B cells are all about producing antibodies against a specific antigen. Clonal expansion is the way that a specific population of B cells undergoes rapid division and increases its capacity to make these antibodies. They're proteins, those antibodies. So clonal expansion results in many, 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 many more of the specific B cells that can respond to that antigen. Now, most of those producing antibodies, and more on antibodies in just a second. Most of the B cells that are produced in clonal expansion go on to produce antibodies. Those cells are called plasma cells, a weird term. But plasma cell is a specific cell type of B cells that originates from clonal expansion that will make antibodies. Many fewer of the B cells that are produced under clonal expansion are stored away in the lymphoid tissues. These we call memory cells. And it's a great term, memory cell, because these B cells, with their antigen receptor, are stored away in the lymphoid tissues in the event that this antigen is encountered a second time. And if it is encountered a second or subsequent time, typically the immune response is much faster and much more robust with regard to clonal expansion. Now, about the antibodies, what do? They're products of B cells. These B cells, the plasma cells, B cells that are producing clonal expansion, start cranking out antibodies. start producing antibodies. Now, antibodies are almost like antigen receptors. That is, there's a portion on the antibody, on the ends, that recognizes a specific antigen. And so, by recognizing it means it will bond to, it'll associate with that particular antigen. Now remember, that antigen is on the surface of a bacterium or a virus. And when that antigen gets bound up by the antibodies, some great stuff can happen. Like clumping can happen. Because each antibody has two binding sites, these antibodies really become linkages between the individual bacteria and hold them together. This makes it easier for the macrophages to do their work, because the antibodies are now contained more or less in a big kind of matrix or mesh of bacteria and antibodies. And so the macrophages can come and do their thing with a big concentrated population of the bacteria. Those antibodies also work to increase the complement activity. They work to increase the macrophage activity. When macrophages encounter antibodies, they get even more excited, more activated. And the antibodies work to, here's my page here, stimulate the natural killer cells as well. So, antibodies extremely important. And again, they're big proteins, antibodies produced by the plasma cells, subset of the B cells, that work in 4 or 5 different ways to very effectively contain, especially bacteria, but sometimes viral infections. All right, on to the T cells. T cells are also part of the specific immune response, or adaptive immune response. T cells, like B cells, have antigen receptors. They have receptors on them for very specific antigens. Now, there's a little catch here, though. T cells, they need the antigen brought to them. Eventually, the B cells are just going to encounter the antigen when it gets into you and undergo clonal expansion. The T cells need the antigen delivered to them. So the antigen gets into you, the macrophage starts gobbling up the bacterium or the virus, and then those macrophages display the antigen. So let's talk about that. Here's a macrophage. It's gobbled up a bunch of bacteria, and those bacteria have an antigen, a large molecule on the surface, that's going to trigger a specific immune response. Let's talk about, again, the T cells. The T cells need the antigen brought to them before they can get activated. So the macrophages gobble up the bacterium, and then they display that antigen, that molecule, from the bacterium on a large complex on their cell surface that we refer to as a self-antigen. And it's called a self-antigen because your cells make it. An antigen because it plays a role in triggering an immune response. The self-antigen belongs to the MHC class. Now, the self-antigen is going to function in 2 different ways. One, it's going to display the foreign antigen, that's the circle on the board there, almost like it would display a trophy on a pedestal. And that self-antigen is going to allow the T cell to dock, like shake hands, with this macrophage. So the T cell comes along, here's my T cell. The T cell comes along, and the T cell docks to the self-antigen and at the same time recognizes that foreign antigen because the T cell has an antigen receptor. And now, when these 2 things happen, the recognition of that foreign antigen in association with the self-antigen, like shaking hands, recognizing the foreign antigen, now the T cell becomes activated. This T cell is going to be either a helper T cell or a cytotoxic T cell. And the helper T cell, once it's activated this way, it's going to start producing chemicals that increases B cell production. It helps that way. Cytotoxic T cell recognizes the foreign antigen and the self-antigen on this macrophage. And that cytotoxic T cell kills this infected cell. The T cells, though, again, need the antigen presented to them. So the macrophages have to display it on the self-antigen. The self-antigen becomes the mantle, the showcase, if you will, the foreign antigen, as well as the contact association molecule for the T cell. Side note about self-antigens. Self-antigens are what are present on all of our cells. These molecules are what we say we try to match with regard to a tissue donor and a recipient. That is, if I needed a new kidney, I would look for a donor whose MHC class of self-antigens is most familiar with, or most similar to my own. This would assure that my T cells don't bump into these new cells on the new kidney and interpret these new cells as being foreign because it doesn't recognize or match up with the self-antigen. And a big immune response often occurs when an individual doesn't fully accept the donated tissue or organ. So they'll reject that tissue or organ. That's because the immune system mounts a big specific or T cell mediated response to that new tissue or cell. So I hope this clears up a little bit about the innate and specific immune responses. We describe them separately. The truth is they are very closely related. You can think of the innate immune response as being kind of the first line of defense, but I can guarantee you as soon as that innate immune response is occurring, the specific immune response, especially if this is a bacterial or viral infection that's occurring a second time, that specific immune response is also going to be activated pretty quickly. Innate immune response, specific or acquired immune responses. Specific or acquired immune responses, the role of the B and the T cells. B cells, antibodies, T cells engaged in what we call cell-mediated immunity. And T cells, remember again, need the antigen presented to them.